concerned. If you have a technology just for rich people, it's never a good technology. So my dream is to decrease this cost from $2 million to $1,000.
Hey there, I'm Luca Fusar-Bassini. I'm a PhD student in computational biology at EPFL in Switzerland, and you're listening to The Biotech Futurist. The Biotech Futurist aims to foster deep understanding and discussion about exciting hot topics in biotech, but I want to say from the beginning that it is by no means rigorous in teaching the subject. And for the sake of outreach, sometimes we need generalizations that, of course, simplify the reality of the science behind what we're discussing. But I can say that my guests and I do our best to be clear and to go in depth. You can imagine to be out with me and my expert guest for a friendly conversation to get a general understanding and more curiosity, having fun as much as I've had recording this podcast. This podcast has no sponsors and any reference is not meant to support any commercial activity. This podcast is a solo effort, so if you wish to support me, I'd be grateful if you followed the Biotech Futurist on Spotify, Apple Podcasts, YouTube, Instagram or your top podcasting platform and share it with your friends. With that said, I am excited to move on to today's conversation at the Biotech Futurist. Welcome back to the Biotech Futurist. Today I'm delighted to host Vittoria Raffa, who was my professor of nanomedicine and molecular biology at the University of Pisa. Vittoria has roots in chemical engineering. She's an associate professor in Pisa and she's been a leader in nanomedicine since a very young age. Vittoria, it's always great to discuss science in detail with you. Thanks for being here. I'd be happy if you could tell us briefly about your journey as a scientist and as a person, from your curiosities and dreams to what you're doing these days.
Thank you, Luca. My pleasure to stay here with you and the other attenders. So talking about my science, I have different interests in science and in research. As my interest is to explore a multidisciplinary field, because as you know, Luca, I have a degree in engineering, but I am professor of molecular biology. So let's say I have the interest in exploring, discovering new technology, radically new technology, and to apply this technology in the field of biology to provide a new solution, a new opportunity with a light to new therapeutic application. Actually, my research interest is in the neuroscience field and also in the gene editing field. Okay, so since the beginning, I was involved in many European projects, trying to establish interdisciplinary, multidisciplinary networking with experts in physical science, material science, photonics, biology, neuroscientist, expert of gene therapy, in order to think to radically new technologies. Surely, gene therapy is one major field of interest. Today, the focus is in CRISPR-Cas9 technology for application in the gene therapy. My journey in this field started a long time ago when I was a PhD student working with nanotechnology, and I was attracted by a special type of nanoparticles, we are carbon nanotubes. And at that time I was, let's say, interested in a question that from let's say, a physical point of view, an engineering perspective, my question was, what happens if these carbon nanotubes working as a small nanoantennas are in contact with neural cells, so neuronal cells? Can I use these small nanoantennas to generate by electromagnetic waves to, let's say, generate electric field? And can I use this electric field to, for example, modulate the electrical activity of neuronal cells, or to perform electroporation in order to transfect neuronal cells with plasmid? And so this was my idea to explore this idea in order to perform gene therapy on neuronal cells, which are hard to transfect cells. And at that time, it was really successful. So starting from this original idea, a few years ago, I continue, let's say, to be interested in this topic. And of course, the idea was a little bit changing because today, when you talk about gene therapy, of course, you talk about CRISPR-Cas9 technology. So I'm still working this field, but trying to exploit the new tools that are available in the field, and especially the CRISPR-Cas9 machinery. But basically, the question remains the same. Can I use novel materials and to exploit the properties, the physical properties of these materials to create smart technological tool for gene editing? And this has a real application, the real world for gene therapy. So this is in brief my research interest, Luca.
Awesome. I guess that this first answer already put us in the middle of your research, so I'd be happy if you could give us a brief panoramic tour in the projects that your lab is currently focusing on, and why do they matter, what is their position in the bigger picture, and what are your dreams for the next few years on your own research?
My dreams is to give a chance for therapeutic success to the concept of programmable nanomedicine. The concept of programmable nanomedicine is a really new concept. So the idea is to use the new tool that we have in the field of nanotechnology and synthetic biology to generate nano tools that can be triggered by external stimuli. Why this is important in medicine and in the biomedical field? Because my dreams is to think to a new generation of medical products that can be safer for the patients. So the general idea of programmable nanomedicine is to have a new generation of therapeutics whose activity can be switched on and off according to the therapeutic desire. For example, just to give you one practical example, we are working on a light-switchable nanoformulation for genome editing based on the CRISPR-Cas9 system. So the idea is to give this as a medication in a not active form, okay? And then you can activate by light this nanoformulation just in the tissue that you need to treat, or in the moment that you need to apply the therapeutics. And so this, let's say, this concept of programmable nanomedicine increases the safety profile of medical products. Because if you administer the formulation, these drugs in a form that is not active, you don't have any side effect. And so you can just activate the therapeutic activity, you can switch on the therapeutic activity when you want, if you want, and where you want. And this, of course, increase the safety, but also the efficacy profile. For doing this, we need to integrate current knowledge in different fields: synthetic biology, nanotechnology, gene therapy, material science, whatever you want. So you should play at the boundary of different disciplines.
That's awesome. So to sum up, one of the crucial questions that we'll be discussing today with Vittoria is the issue of delivering nanomedicines in the right place, so we don't elicit off targets and undesired effects at other organs in our body. But this is hard to do, especially if you only focus on molecular aspects of these tools, as we have to really recognize subtle cellular differences and only target the disease, let's say, cells. So one possible solution that Vittoria is exploring is to, administer these nanomedical therapeutics whole body, but then activate them only when and where they are needed by means of some external means, such as light. And this is the idea that Vittoria was describing in the iGene project. And I'd be happy if you could discuss with us a little more in detail what are the nanotransducers that you're using in this project, and why they constitute a safer tool for nanomedicine compared to a state-of-the-art, especially as what you've called the when, where, if functions integration?
Yes, thank you, Luca, for this question. So, um, as I told you before, the idea is to put together different concepts that comes from synthetic biology, nanotechnology to generate this stimuli responsive nano platform. I think that we can call some concept that comes from logic gate, okay, to understand how we can benefit from this integration. So think a little bit to the Boolean algebra. So the logic gate, the truth table, no? In this context, if you think to the CRISPR-Cas9 system, the CRISPR-Cas9 system could be understood as one input logic gate. And the output is the gene editing. And the gene editing comes true if the input comes true. In this specific case, the input is the recognition of the target by the guide RNA. Okay, so if you think in this, a lot of this consideration, the CRISPR-Cas9 could be considered one input logic gate. The output gene editing comes true if the input, which is the guide recognition of the target, comes true. Well, our general idea is to implement multi-input and logic gate, which is the advantage. The advantage of multi-input and logic gate is that for the output to come true, more than one input, 2, 3, 4 different inputs should simultaneously come true. And so by doing this, you can increase the safety of your treatment, because if you increase the inputs required for the output to come true, this means that you strongly reduce the off-target events. Okay, and so which are these inputs? So we are playing a lot with this. For example, light could be one additional input, or additional input could be the recognition of different sites. For example, in order to have the gene editing event, the simultaneous recognition of two different alpha sites should happen. So you have input number one, recognition of the first alpha site onto the target. Second event, second input, recognition of the second alpha site. Third event, light on. So in this case, you have a 3-input at logic gate, and so all these 3 conditions need to come true to have the output. So this means that by using this concept of synthetic biology, you can realize a triggerable, a switchable system. And so you can have your therapeutic activity, which is the gene editing activity, when all these conditions are going to happen, okay? And so this is something that simultaneously can increase the safety of the treatment, but also the efficacy, and also reduce the side effect, because you are targeting just the tissue. Okay, in this specific case, the tissue irradiated by light. Any other tissue is not irradiated by light is not interested by this activity. And so, which is the big advantage? The big advantage is that you really expand the therapeutic application of gene editing, gene therapy based on the CRISPR-Cas9 system. So this is our, let's say, long-term dream.
That's awesome. So to summarize, the idea is here that by combining different inputs, you can reduce unwanted activation of the system, because it's highly unlikely that all the 3 or more of them will come true simultaneously if you don't have the conditions that you really want to target, whereas a single input is much more likely to turn true when you don't want it to turn true, right? So I think this really puts us in the middle of a conversation on today's episode, that is DNA editing techniques, safe and effective nanomedical delivery, and in general scientific discovery and innovation where fields intersect. I guess we can start from gene editing techniques, specifically focusing on context-dependent advantages and limitations of each technique, because each technique is not in itself perfect or imperfect. Each technique becomes useful when you set the right context. So first, I'd be happy, Vittoria, if you could briefly describe for us how CRISPR-Cas9 techniques have completely revolutionized how we do science in basically almost all biological fields, I suppose. Based on your personal experience, you were already an established scientist when the CRISPR revolution happened. So how did the field react when the first papers came out? How was your reaction, the reaction of your colleagues? Was there skepticism or enthusiasm when you first tested them? Were you feeling that it worked, or were you skeptical about it? I'd be very curious on the human also side of the equation of this substantial revolution in the biofields.
Well, my reaction, I guess, was similar to the reaction of all molecular biologists in the world. We really realized that this was the beginning of a new chapter of the biotech and molecular biology history. Thinking to my reaction, well, at that time, I should say that the concept of tools for editing genomes was not totally new. Let's say at that time there was other tools, finger nucleases or TALEN, to perform the genome editing of living organisms. But with the discovery of the CRISPR-Cas9 system, that was really clear for me that it was a breakthrough. Why? Because I really understand that this new technology has a huge therapeutic potential. And this was not the same for the previous one. So the previous one technology was really outstanding for a biotech, but with the discovery of the CRISPR-Cas9, that was clear to the scientific community that we can expand the application from the biotech to the therapy, so gene therapy field. So that was my first reaction. We have the tool to think to how to modify human genomes for therapeutic application. This was the first reaction. The second reaction was, okay, this tool is so much powerful that we as a scientist should, let's say, take into account some ethical concerns. So we should be responsible in the use of these tools because really we have a big power and a lot of new knowledge in a very short time frame that we really need to understand which are the potential but also the limitation of this technology. Not to stop the discovery, not to stop the science advance, but just to understand which are the limitations and how to overpass this limitation, thinking to the therapeutic application. That's my first and my second reaction.
So I guess the expectations were set pretty high since the very beginning of it, right?
Absolutely. That was clear since the beginning.
That's interesting, yeah. So what about some broad definitions for gene editing technologies? I guess most listeners of this podcast will be familiar with CRISPR-Cas, as it's one of the very main protagonists of these episodes. But maybe we could spend a couple of words to discuss a little bit about base editors, prime editors, base editors, CRISPR-I and CRISPR-A for transcription repression and activations, the last two of them. So these are all techniques based on CRISPR-Cas, but they have different features that make them pretty useful in different situations. Could you describe them for us a little bit, Vittoria, and also compare them? So what's the advantage of the one, what's the limitation of the other one?
Well, that's not totally clear to the scientific community. We are still exploring the limitation of this technology. Just to have a very, very short summary on them, and we can, let's say, discuss more on specific subject if you want to look up. But just to give you a summary. Well, the traditional CRISPR-Cas9 system can use different Cas variants. The most popular is the Cas9 variant. That is a nuclease that is able to perform a double-strand break. And this double-strand break can be used to knock out or knock in the gene expression, depending on if you have or if don't have a donor template. Well, a limitation of this technique is that at low frequency, It's also possible that these enzymes recognize off-target sequence that are similar to the target, but let's say they differ from 1 to 3 bases in the target sequence. This off-target could happen at low frequency, but it's still possible that the enzyme recognizes this off-target sequence. How you can solve this? Of course, it's possible to select variants that are high fidelity variants of Cas9 system. Well, there is another system which is based on a mutation of the Cas9 protein, which is the dCas9, catalytically dead Cas9, which is used to perform interference. What this means? Just to give you a simple example, when you want to express one gene, what should happen is that there is some chromatin remodeling, and onto the promoter, you need the assembly of the transcriptional machinery, the pre-initiation complex, for example. So if you have one enzyme, okay, this catalytically dead Cas9, which is able to bind to the promoter, but not to cleave, just to bind on that, for a sterical hindrance, you are blocking the assembly of the transcriptional machinery. So you are knocking down the expression of that gene. Okay, but you can use the same system to activate the expression. For example, you can create a recombinant protein. Okay, so for example, you can express Cas9 as a recombinant, the catalytically dead Cas9 as a recombinant protein, and you can fuse to the C-terminal or N-terminal of this catalytical dead Cas9 one co-activator of the transcription, one coenzyme which is a positive, for example, modulator of transcription, or could be a chromatin remodel or a modifier of the chromatin. So in this specific case, you can target this system close to the promoter. Then you can bring onto the promoter this positive modulator of the transcription or chromatin remodeling, and so you can activate transcription. Okay, by using this concept to produce the Cas9 as a recombinant protein, you can, let's say, fuse to the N-terminal, C-terminal, the Cas9 protein, also other domains or other protein, for example, the base editors. Okay, so by doing this, what happens that in specific loci into the human genome or genome of living organism, you can target base editor, and these base editor are able to perform a single base mutation. What this means? This means that you have the red pencil to rewrite the genome by using a single base change, and this is the concept of base editor. Or, for example, with the prime editing, you can, let's say, use a reverse transcriptase, so to fuse to the Cas9 the reverse transcriptase, and you can also modify the guide RNA to include in the guide RNA a guide template. The reverse transcriptase can use to copy a small strand. Okay, so insert small sequence into the target point. This editor is really new. That's really interesting. The concept is like the prime editor, but what you are doing is to insert in the target locus integrase site. And so you can couple this with serine integrase to perform, let's say, in a smart way, gene by combining this serine integrase with the prime editing. I guess it's too much, is a lot of concept in few minutes. But the point is that you really have a molecular scissor, and by this molecular scissor you can cut, copy, paste, modify, edit any sequence, whatever you want, where you want. That's the point. You understand that the number of possibility that we have is uncountless.
So basically this tool has enabled scientists to bring almost everything to DNA, right? So ranging from enzymes that edit specific bases, Vittoria was mentioning this thread pen to change the letters of DNA one at a time, and other enzymes that can modify DNA specifically to activate transcription or inhibit transcription rather than modifying the letters themselves. So this opens a full range of opportunities for different disease settings. I'd be happy if now, Vittoria, you could explain to us a few examples of disease settings in which CRISPR is relevant, specifically now. I mean, in the long run, maybe it will be relevant in almost all disease settings, as acting on gene expression or genes themselves can really prove therapeutic in a number of contexts. But for now, what's achievable? What are the criteria to understand what's achievable, and which diseases can really be amenable in a feasible fashion with the technologies that we have now?
Oh, well, this is a very, very difficult question. And we can talk, let's say, hours and hours about that. So, well, there is something that is feasible now, something that will be feasible in few years, and other things that are feasible in 1 decade, in my opinion. So let's start from the present. Okay, what we can do now is to use the CRISPR-Cas9 system to treat genetic disease, but also cancer diseases. Well, I want to give you some numbers, Luca, just to give you an idea. The number of the ongoing clinical trials worldwide that use this system for gene therapy is 26. Okay, that's a huge number, but not so big if you consider the plethora of different diseases that you can potentially target. Okay, so we should ask ourselves why we have this relative small number of clinical trial. Point is that it has been estimated that the cost of this therapy, if I'm not wrong, there are different estimations, but the cost is $2 million per patient. And so what this means, I think this is the real important question that tell us where we are. Okay, we are in the stage that we have very powerful tool, but it's very, very expensive. And so this raises also ethical concern, because we are developing a technology that is available for all, or just for rich people. What about the developing countries? Which is the direction that we want to go to this new science. The point is, why is it so expensive? I should say there are a lot of reasons, but the most important one, probably the most important one, at this stage, you need viral vectors to deliver the CRISPR-Cas9 machinery. Okay, there are also alternatives, for example, physical means that is based basically on electroporation, but this also applies to the ex vivo paradigm. What this means that you can harvest the cells from the patients, you edit the cells, then you reimplant the modified cells into the patients. But also in this specific case is not very popular. So the use of viral vectors is the preferred one. And this is really, really expensive. Okay, and so we have, yes, one tool for all care diseases, but it's not affordable for the national healthcare systems. So in my opinion, the direction, it's the needed direction, the direction also we need to go is to ask ourselves, how is it possible to decrease this cost in order to make this technology available and affordable for the national healthcare system? And this brings me to my research, because surely nanotechnology, nanomedicine could give the chance to decrease the cost and to expand the therapeutic application. Okay, so I should say we should invest in the use of non-viral alternatives for delivering the CRISPR-Cas9 machinery. Okay, in my opinion, this is, if we can, let's say, be successful in this strategy, in the increase of therapeutic application will increase exponentially. And also we should, let's say, solve the problem of increase the safety profile of the treatment. And this brings me to the second part of my research line, which is to make this system stimuli responsive, or to activate the activity, to trigger the activity of this formulation in order to avoid the side effects. Okay?
Yeah, this is insightful and clear, thank you. So I guess I may ask you about the second thing that you mentioned, that is, what about in 10 years from now? Of course, accessibility is the number one challenge, and it was a captivating explanation, and also how you introduced to us nanotechnology. You know, viruses need cells to be made, so they cost a lot of money, probably also because, for making them, you need to have cells and keep cells in a proper bioreactor and so on. So nanotechnologies based on chemical tools help the scalability. So what do you suppose may happen in the next 10 years, as you were briefly mentioning?
Well, in the next 10 years, I guess we have the CRISPR-Cas9 system literally can be used as a tom-tom to guide any medication to the target blocks. Okay? Just to give you one example. So 10 years ago was a very popular to use inhibitors of epigenetic modification, blocker of epigenetic modification. So medication that block acetyltransferases activity, or methyltransferases, or whatever you want, the methylase, whatever you want. And this was a really popular last decade, let's say, 10 years ago. But at that point, what the scientists realized is that it's toxic. Because, of course, if you block epigenetics modification, this is something that is related to the whole genome. And this is really toxic. It's not just related to the genes that you are interested in, okay? And so this failed. But what they expect from the CRISPR-Cas9 system, that it could be a system to reduce the toxicity. Because you can really use this system to target all this medication, not to, let's say, a random lodge, but a specific locus to regulate the gene expression, the specific lodge. Okay, so what I expect in the next decade that we can modify and modulate the gene expression at any level, epigenetic level, transcriptional level, post-transcriptional level. So we can use the system to modulate as we want the gene expression. And you understand that this applies to any disease, any treatment. So that's my expectation, that we can have a tool to gain control on the modulation of gene expression, which is a site-specific. Okay, how the nanomedicine could contribute to this? We have an extreme need of carrier that are able to bring this machinery to the precise target in the right tissue, in the disease cells. And if we don't have such drug delivery system, of course, any treatment will be not effective. And we can also use nanotechnology and synthetic biology to make this system stimuli responsive. And we can use different stimuli, light, electromagnetic field, temperature, pH, whatever you want. Also drug responsive, ions responsive, microRNA responsive. There are a lot of stimuli that you can use to switch on the activity of this system in the tissue that you want, when you want, if you want, where you want. So this is the where, if, and when integration function.
Insightful. Yeah, the CRISPR promises are pretty high, as we were already discussing with Vittoria and I in the inaugural episode of this podcast, and I'm looking forward to discussing with you in a few years what's happening. So I guess this leads me to another very big question in the field, that is the immunogenicity of gene editors. How can we measure, predict, and control if a given gene editor and the nanoformulation that carries it elicits some sort of immunogenic adverse effects or reaction that may not only result in a loss of the desired effect, but also in bigger problems, and at some point the impossibility to repeat treatments if these immunogenic mechanisms set out at the first round of treatment, right?
That's true. I'm not really an expert on this field, Luca, but as far as I know, the problem is what you explained, which means that the problem is the loss of efficacy and also the reaction, immunoreaction. So as far as I know, scientists find anti-Cas9 antibodies in the human serum. And so we know for sure that's a big issue that we can solve. The good news that we can use the knowledge that we acquired in the field of humanized antibodies, and we can use this knowledge. We can take advantage of this knowledge for also in this field, in the genome editing field. So, as far as I know, there are some technique for masking epitope, immunogenic epitope. There is also the opportunity to select a variant. I told you before that there is this new variant. For example, you can use non-pathogenic bacteria to select this variant. This could be another option, as far as I know. The last, let's say, is not the best, but also another option is to use immunomodulators when you co-administer this medication. Well, I don't want to go in depth. It's not really my field. But for sure, Luca, I know that there is a lot of knowledge already acquired in the field of gene therapy and also monoclonal humanized antibodies that we can reuse. We can take advantage of this knowledge to provide a new solution also in this field.
Mm-hmm. This leads us back to the idea of merging different fields to gain knowledge.
Absolutely.
I guess my next question would be, what technologies would you love to exist out there to speed up and make better, in some sense, the process of developing a gene editor? What will be, for instance, the role of AI-based protein design and optimization, and of metagenomics-derived tools? And by metagenomics here, I mean the discovery of tools, in this case proteins or ribonucleoproteins, such as the CRISPR-Cas systems from microorganisms that are rich in these tools for the most disparate functions, such as, for now, people have found some tools for methane metabolism that may turn useful. The Duad now, the founder of CRISPR field, is in this study, and there's lots to discover in metagenomics. So people, as you've already mentioned a few times in this episode, are now bringing to DNA several different enzymes, ranging from epigenetic modificators to co-activators of transcription and so on. So I guess another question that I have for you, and I know it's a lot of questions together, is what enzymatic function would you dream to bring to DNA, something that hasn't already happened, and why? So to sum up, my questions here were, if there is some technology that you'd love to exist out there to speed up and make better the process of developing and making a new gene editor, what do you think about AI and metagenomics derived tools? And third, what enzymatic function would you love to bring to DNA and why?
Wow, that's our 1 million question in one. I really believe that it is strategic for the field to use a synthetic biology approach. And artificial intelligence it will be crucial for the selection of new variants and to predict the behavior of the new variants. It's something that you can do also without artificial intelligence, the selection, but you understand that time scale of this process is decades, and we don't have any time, because you see, every 6 months there is a new paper that shows another possibility, so we don't have such a time. Every month is say time for a new discovery, and so in my opinion, artificial intelligence could help us to predict the behavior of new variants, and we can, let's say, use this to understand which is the best synthetic biology approach for generating this new variant. Just to give you a practical example, you can create new variants which are high fidelity, or they can recognize different bump sites, and so you can expand, let's say, your tools of nucleases. Okay, and for example, you can create variants. For example, you can split. The name is a split Cas9. You can split the N-terminal and the C-terminal, okay? And you can superimpose that there are the reconstitution of the full-length Cas9 under specific conditions, okay? So it's really interesting, in my opinion, to use artificial intelligence to predict under which condition you can have the reconstitution, the full length. Because you understand that this also recall the concept of the AND logic gate. Because if you can have this reconstitution of the full length activity just in specific situation, this means again that we have a molecular schizol which is not active, but that activate under specific condition. So in my opinion, there is a lot to do to select new variants that have this superior behavior that you can control into the target cells. So this is very interesting and stimulating field, in my opinion, and I expect that we'll investigate this in depth in the next years.
Awesome. So I guess it's time to discuss a few concepts of nanomedicine more in depth. You've already introduced us to several of them, including the idea of making nanomedicine programmable and Q-responsive. And by Q-responsive here I mean really making nanoparticles that can deliver their cargoes by responding to specific stimuli of several different types, as Vittoria mentioned, such as metabolites or enzymatic activities, pH, redox state, temperature, light, and so on, up to the given sequences of mRNAs that she was mentioning, or other RNAs and DNAs to unlock cargoes such as DNA origami. So the full spectrum of possibilities is as big as the one of engineering CRISPR and Cas-derived systems, so to make novel tools probably. And by combining the two of them, probably we will find incredible tools for different contexts, different pathological contexts. Of course, each of them will be more suitable for a given area, and each of them will be less suitable for another area. So clinical applications will be initially at least targeted to where we really believe that this can work, and I'd be happy to explore with you, Vittoria, now a number of current nanomedical tools that are being explored in clinical trials and preclinical mouse models. And for each of them, I'd be happy if you could briefly mention to us the main advantages and if there is either a clinical application that you are aware of or preclinical evidence that could lead to clinical trials in the foreseeable future. And also, in general, if you can mention a few trends that you see for this technology, if it's a technology that we really have to keep an eye on. So maybe we should start from viral-like nanoparticles, and you've already mentioned adeno-associated viruses and the difficulties related to making these technologies accessible, as they cost a lot of money. There's also a clinical trial going on here that I've learned more by reading a review from you and your colleagues. So I'd be happy if you could discuss also this clinical trial to introduce the idea of viral-like nanoparticles.
Okay. Well, yes, probably it's better to start from clinical trials. When I tell you that nanomedicine could really help the field, it's not just my opinion, it's a matter of fact. Before, the large majority of ongoing clinical trial are based on the ex vivo paradigm. Okay, what this means that you are editing the cells from the outside and then reimplanted the cells inside. If you think to add application, another application, for example, is the treatment of the eyes. And the treatment consists in the subretinal injection of adenos-associated viral vectors. Okay, but you understand that this is not a real in vivo. It's let's say a local treatment. As far as I know, there is just one case of real in vivo application, which is based on the use of a nanoformulation. This is a lipid nanoformulation that was designed to treat transthyretin amyloidosis. So just a few words about this pathology, Luca. So transthyretin is a protein that when is mutated is not able to tetamerize, but is misfolded. And so it deposits in the cardiac tissue in the form of amyloid fibrils. And so this causes cardiac failure, and so the half-life expectancy of these patients is more or less 5 years. So what the scientists did is to design this nanoformulation, the CRISPR-Cas9, to perform the knockout of transthyretin, mutated transthyretin. But the novelty of this approach is that by injecting this lipid nanoformulation, what happens is one mechanism that is called opsonization. So after injection, the nanoparticles are coated by opsonin, and more specifically by ApoE. Okay, so ApoE protein has specific receptors that are enriched onto the surface of hepatocytes. So this means that you are injecting systemically this nanoformulation, but it accumulates just in the hepatocytes, which is the sites of production of transthyretin protein. What we are doing is to use this nanoformulation and so nanotechnology to make an in vivo treatment a local treatment. And this increases the safety profile of the treatment because we are targeting our CRISPR-Cas9 machinery just into the hepatocytes, so just in the target tissue. So this is the big potential of nanomedicine, to target treatment. This is just one opportunity. You mentioned DNA origami. By using aptamers concept, you can, let's say, open this cage to release their content, in this case, the CRISPR-Cas9 machinery. One really nice application is to use microRNA as a ligand to bind to the DNA cage and to trigger the opening of the cage. Why this is interesting? Because some cancer cells have an overexpression of specific microRNA. So the idea is that you can inject this DNA origami that are carrier of the CRISPR-Cas9 machinery, and when these DNA cages are internalized by these cancer cells overexpressing microRNA, the cage open and release the CRISPR-Cas9 machinery. And of course, this is a stimuli-responsive nanoplatform. One very interesting is the exosome vesicles. Okay, why it's really interesting? Because you can create this exosome vesicle by using as starting material some specific cell type, also patient's-derived cell type. So you can harvest these cells from the patients, so you can dissociate the cells in order to recover the phospholipids, and you can use this as starting material to build your exosome vesicles. There is a sort of trophism, so by doing this, you have vesicles that retain a memory from that kind of cells. And so by doing this, you can target your medication to that specific cell type that are in the memory of these exosome vesicles. This is also another interesting application. The most interesting to me is the use of the golden nanoparticles, because these are the subject of the iGene project. And why it's really interesting to me? Because, we make some preliminary experiments with the use of golden nanoparticles, and we really found very interesting results. We understood that these gold nanoparticles are able to work as a carrier of the CRISPR-Cas9 system into the cells. And so when the ribonucleoprotein is functionalized to the gold nanoparticles, they are spontaneously able to cross the cell membrane, also the nuclear membrane, and to deliver the cargo into the nuclei. And another interesting feature is that they are plasmon nanoparticles. So you can irradiate them with a light, and you can activate the behavior of these nanoparticles as a nano eaters. And we want to use this behavior to induce locally in a specific point in the genome, the target cells, local DNA melting. Okay. Why this is important? We are using this approach, Luca, to provide a new medication for treating melanoma. Our idea, by using this approach, is to target the repetitive loci into the DNA, in order literally to mince the DNA of human melanoma cells. Okay. We want to kill these cells. Melanoma disease, one problem is the multi-drug resistance. And there is also the problem that because of this multi-drug resistance, they can escape. Okay? They can generate resistant clones. With our approach, if we target the repetitive loci, DNA loci, what we can do is to totally destroy these cells. The idea is to create this light-responsible nanoformulation in order to totally destroy this skin lesion by applying this as a nanoformulation by topical application onto the lesion site. So this is our idea, more or less.
Okay, so I guess now it's time for my more speculative questions. I have two questions. So my first one would be about mitochondrial disease. As many of you may know, mitochondria, let's say evolutionarily very, very far away in time, come from bacteria. So let's say that they preserve their own very small DNA. So mitochondrial disease can arise sometimes due to issues in the nuclear DNA, but sometimes also due to issues in the mitochondrial DNA itself. And this raises the possibility of performing mitochondrial gene editing, and this is something that has already happened. I guess 2 years ago, a beautiful paper by the group of David Liu, who was also the group that invented the idea of base editing and at least prompted this idea to a neat optimization. They also created this first base editor for mitochondria. So I guess that making gene editing happen for mitochondrial disease will also open a few more challenges. What are the potential hurdles in delivery and editing for mitochondria, and how are current strategies working for this, and their limitations? What do you envision in this field?
As far as I know, one big problem is that relating to the mitochondrial genome, we have many copies. Okay, and in order to be therapeutic efficacy, you should target a certain number of these copies. Should say 20% at least, 30% at least. So the problem in the field is the delivery of this machinery inside the mitochondria. I think that there is a big potential in the field of nanomedicine to solve this. Okay, for example, when we performed experiments in the framework of iGene project, what we found is that gold nanoparticles are really very useful in localizing the CRISPR-Cas9 machinery into the mitochondria. We performed some transmission electron microscopy, and we found that a huge number of nanoparticles are localized in the mitochondria. We really don't know why and which is the mechanism, but they can cross the mitochondrial membrane. And so, of course, in my opinion, they can provide new solution and really give new opportunity to use all the tools that we have, base editor, prime editor, base editor, whatever you described, also to the mitochondrial diseases.
Yeah, that's interesting. So I guess now my very broad open question that I typically ask to my speakers is, can you tell us about what you envision in the world, in this case of CRISPR and RNA therapeutics in the next 10 years? And specifically, I'd be happy to know, will it be fragmented into more and more startups? And if this is the case, what will be their relationship with one another? And you've already mentioned a lot about how regulations and accessibility are a big part in this field. I mean, the technologies are there and the optimization will make incredible tools, but we have to make sure that they are scalable and accessible, and lots of things that do not concern the scientific findings themselves. How do you see the regulatory landscape to introduce generators to the clinic from the perspective of someone who lives in Europe? As we've already discussed a bit of this for people living in the US. Do you think this is something that is already happening in some parts of the world or will happen in the foreseeable future? So to sum up, I think I've asked you a lot of questions in one. I'd be happy to know about your view for the CRISPR field in the next 10 years, with details also on the regulatory and the startup picture, and not only on the science of it.
Yes, the point, in my opinion, is try to make this technology accessible to the population. Okay, so for sure, we know that we have this nuclease and we can use them in any situation to modulate the gene expression. For sure, we have a number which is limited for now of clinical application, but in, let's say, 5 years, these numbers will exponentially increase. The point is the cost. As I told you before, it's millions of dollars per patient. So this means that we are going in the direction of insurance systems that can cover this cost. Technology not available for all. And this is an ethical concern. If you have a technology just for rich people, it's never a good technology. So my dream is to decrease this cost from $2 million to $1,000. How this is possible? This is possible, in my opinion, with non-viral vectors. Just to give you an idea, gold nanoparticles, the raw cost of our gold nanoparticles, just to have the quantity for treating one patient, is €1. So you understand, if your raw cost is €1, you can add the cost of the ribonucleoprotein, or to produce this in a clean room, to produce this under controlled condition. It will be thousands of euros, not millions of euros. Okay, and so that should be my dreams. From the biological point of view, we have the tools. Now we should use also other field, engineer-like field, try to make these tools economically sustainable. That's the point, to have expansion of the therapeutic application. But this will be possible if these therapeutic are sustainable, economically speaking. Also in developing countries, there is this, let's say, it's mandatory in my opinion, to give the priority of those technological solutions that can be also used in developing countries. So this should be that direction. To have one tool that is the all cure disease for all the people worldwide. So that's my dream.
Awesome. I guess you gave us a very detailed and inspiring picture about why gene editing, mating, programmable nanomedicine can really be the start of a trustworthy friendship. So thank you so much, Vittoria, for spending some time with us today discussing all about nanomedicine and gene editing. I guess there's much more to discover about these topics, and we'll make sure to post the links in the description. So thank you again, Vittoria. It's been a great conversation.
My pleasure. Thanks a lot, Luca.
You've just listened to A Biotech Futurist, a podcast by Luca Fusar-Bassini. This is the first series, and a new episode is out every Monday. Please consider subscribing and rating the podcast on Spotify, Apple Podcast, YouTube, Instagram, or your top podcasting platform. And if you liked this episode, consider sharing it with your friends, as the growth of new podcasts relies on word of mouth. If you have any suggestions, don't hesitate to reach out to me on Instagram or Gmail, at thebiotechfuturistpodcast@gmail.com. You can find the full AI-generated transcript of this episode on my website, lucafuterabassini.com. I'll also post the links to the main papers referenced in this episode, which you can find here in the description too. Thanks for listening to A Biotech Futurist. I am looking forward to talking with you in a week.